As far as the antimicrobials is concerned, and in the treatment of infectious diseases, it's all about selective cell death. Those agents that kill or inhibit growth of microorganisms are often known as antimicrobials. Previously, the term antibiotic was reserved for describing substances that produced by microorganisms that killed or inhibited the growth of other microorganisms. However, now the term antimicrobials and antibiotic and antibacterials are used interchangeably. Basically, if you look at the mechanism of action of the various antimicrobials, you have the cell wall synthesis inhibitors, which are probably the oldest of the groups of antimicrobials, such as the penicillins. You have those that inhibit protein synthesis, such as the aminoglycosides and the macrolides, and then you have the RNA polymerase inhibitors, such as the rifamycins. The quinolones inhibit DNA gyrase and DNA topoisomerase, and sulfonamides and trimethoprim inhibit folic acid metabolism. The selection of an antibiotic is pretty much dependent on the type of pathogen or the gram staining, the site of infection, the route of administration and the dosing, resistance, which is a global problem, allergies, particularly to penicillins, toxicity to certain antimicrobials, drug interactions, particularly interactions with the cytochrome P450 system, the cost factor. The antimicrobials are usually classified in two groups. One is the bactericidal and the other is the bacteriostatic. The bactericidal antimicrobials kill all infecting organisms, and the bacteriostatic ones basically slow down the growth of the bacteria. The first one is the cell wall synthesis inhibitors. These are often known as the beta-lactams, or the penicillins are the typical example, and then carbamazepines, monobactams, cephalosporins particularly. The cell wall synthesis inhibitors, example penicillin, they exert their action by binding to a penicillin binding protein on the cell wall surface, and once they bind to the penicillin binding protein, they release autolysins, which are carboxypeptidases and endopeptidases, which prevents cross-linking of the cell wall. This includes the natural penicillins, the anti-staphylococcal penicillins, the aminopenicillins, and the anti-pseudomonas or the extended spectrum penicillins. Penicillin G and penicillin V are considered to be the oldies, and they are one of the oldest of all the penicillins. And then you have the anti-staphylococcal, the aminopenicillins, and the extended spectrum. As far as the penicillins are concerned, penicillin G particularly cannot be given orally, so therefore it has to be given parenterally, and penicillin V basically has to be given 2 to 3 hours before or after a meal because it is extremely sensitive to the pH of the GI tract. The mechanism of resistance that happens to the penicillins is typically beta-lactamase production by Staphylococcus bacteria, inactivation of the drug, reduced permeability, and the expression of the P-glycoprotein that may pump the antibiotic out of the cell, and also increased DNA repair enzymes, which typically happens with the fluoroquinolone. So the classification of the penicillins, the anti-staphylococcal penicillins are nafcillin, oxacillin, dicloxacillin, and basically methicillin. These agents are basically used in the penicillinase resistant penicillins. They're used in beta-lactamase producing staphylococcal infections. The staphylococcal infections basically produce beta-lactamase, which breaks down the beta-lactam ring in the penicillins. They're mainly used for staphylococcal infections, gram-positive, and staphylococcal or MRSA, endocarditis, bacteremia, and cellulitis. You do find combination products such as ampicillin plus sulbactam and amoxicillin plus clavulanic acid. The extended spectrum penicillins cannot be degraded by a majority of beta-lactamases, and they're used for greater gram negatives and are considered to be broad spectrum antimicrobials, and also for anaerobes, however not for the typicals. The next group group of the penicillins are the cephalosporins, and the cephalosporins are classified into 4 generations. It must be made clear that all 4 of them have gram-positive activity. They do not lose their gram-positive activity as you go up in the generations. However, as you go up in the generation, they gain more gram-negative activity. The other thing about the cephalosporins is that they are much more stable to gastric pH compared to the penicillins. Their structure is such that they are more stable and therefore are less likely to be degraded by the gastric pH. The generation predicts spectrum of activity. As you go up in the generations, the gram-negative activity increases. However, the gram-positive activity. The first generation cephalosporins include cefazolin, cefalexin, and cefadroxil. They basically cover the gram-positive streptococcus, staphylococcus infections, MSSE and MSSA, and they are very limited to their gram-negative activity, almost no gram-negative activity at all. As you move up into the second generation cephalosporins, you have cefoxitin, cefotetan, cefuroxime, which are the more common ones, and cefamendol. They basically increase their gram-negative coverage, therefore they have an expanded gram-negative coverage, and as a result they will cover H. influenzae, E. coli, basically Neisseria gonorrhea, Proteus infections, Klebsiella, and Catarrhalis. They are also effective against, particularly cefoxitin is effective against Bacteroides fragilis and a number of other anaerobic organisms. It must be noted that particularly cefotetan contains N-methyl-tetrazole in its side chain, and it is the NMTT that may cause the disulfiram type reaction if it is taken with alcohol. The third generation prototypes are cefoperazone, cefotaxime, and cefodoximine, and ceftazine. They basically have a greater coverage of the gram-negative infections. They have extended gram-negative coverage, and also including Salmonella, Shigella, and Serratia. They are typically or clinically used to treat infections caused by gram-negative bacteria, particularly hospital-acquired infections or complicated community-acquired infections that may involve the respiratory tract, soft tissue, the blood, and urinary tract and intra-abdominal infections. Ceftriaxone particularly has NMTT on its side chain and may cause disulfiram reactions. The fourth generation basically is reserved for serious in-hospital systemic infections, and the only one is cefepime, which is given intravenously. They are usually reserved for resistant infections and serious systemic infections such as bacteremia, pneumonia, febrile neutropenia, intra-abdominal infections, and meningitis. Vancomycin, which is also considered to be a cell wall synthesis inhibitor, however, it does not bind to penicillin binding protein. Vancomycin actually alters a structure in the chain of the wall. It binds to the muromyl pentapeptide component of the cell wall, and therefore it changes the structure. Vancomycin is a drug of last resort because of its side effects. As a result, vancomycin is used when other drugs do not work because of the typical side effects. It is effective against streptococcus. It is effective against staphylococcus and staph aureus. And basically it is used to treat serious systemic staphylococcal infections, including MRSA, streptococcal, and enterococcal infections. As I said, it's the antibiotic of last resort and is also considered for the treatment of Clostridium difficile that causes pseudomembranous colitis. The big problem associated with vancomycin, which is an infusion-related reaction, is often referred to as the red man syndrome. And this results in fever, chills, histamine release, and a lot of rashes and redness. So you can decrease the infusion rate, lower the dose, or pre-treat the patient with